So I'm going to turn things over to Vaughn Cooper from CBAM. Is that how you pronounce it? Do you call it CBAM or?
We do.
Let's call it. Okay. I'm going to turn it over to Vaughn to introduce our speaker. Thanks for joining us.
Thanks, Charlie. Thanks, Rebecca. It's good to see so many familiar faces. I'm delighted for this opportunity to introduce one of our most valued CBAM colleagues, a co-founder, and really, an engine, a catalyst herself for a lot of our initiatives. In particular, our educational program, which involves I think, fairly innovative certificate in evolutionary medicine within our School of Medicine PhD programs. Anne-Ruxandra Carvunis, she joined Pitt not long after I moved here, and the sort of the ideation of CBAM was really much driven by conversations with Anne and later on with the group that she's built. She's a really model mentor and leader in thinking about evolution in medicine within education and sort of career development. Anne herself has won a number of awards for her pretty pioneering work on the question of where do new genes come from. These include a Searle Scholar Award, an LHAS Trailblazer Award, a NIH Director's New Innovator Award, and a Sloan Research Fellowship. She's also, I'm sure, missing other awards too. We're really delighted to have Anne-Ruxandra and our group at CBAM, and really looking forward to our work. Take it away, Anne.
Hi, thank you, Vaughn, for this really very kind introduction, and thank you to the Club EvMed for inviting me to share some of my thoughts today. I really want this to be a conversation. I prepared slides that I will show and discuss with you with very little data, more to show you some of the concepts and how they're pushing the field of molecular evolution forward in some ways, but also posing some conceptual problems that I would love to have your opinions and help and ideas on how to move forward with that in the discussion part of the presentation. So let's see if I can share my screen again. Can you see my screen?
Looks great.
Okay, perfect. So yes, the title of my presentation is Physiological Implications of De Novo Emergence in Health and Disease. The motivating question of it all is really what makes us different from other species. And it is widely accepted and understood that the evolution of genes underlies the difference between species, populations, and even individuals in health and in disease. How does this evolution happen? Well, through the evolution of species, which is through the process of descent with modification that is symbolized by the tree of life. From some very ancestral origins, species evolve from each other and diversify along the way, resulting in this huge set of biodiversity of extant species that are alive today on Earth. And at the molecular level, it's kind of the same. There's a molecular process of descent with modification that leads to new genes that derive from ancestral genes. The mechanisms of such gene descent with modification are symbolized on this slide. You can have a gene that accumulates a lot of substitutions such that its sequence is now unrecognizable and it's effectively a new gene. You can have the creation of a new physical gene through gene duplication. Two different genes can fuse, a gene can split, and genes can also integrate our genomes through cross-species transfer, making a gene that is new to us in our genome. De novo gene emergence is not that. De novo gene emergence is the process of the transformation of a non-genic sequence into a novel gene. A gene that did not descend by descent with modification from an ancestral gene, but arose in some ways from scratch. And that current estimations suggest that it may be the largest source of orphan protein coding genes in eukaryotes. And by orphan protein coding genes, we mean protein coding genes that have sequences that are unique to one species or one lineage to the exclusion of all others. We cannot find gene families for them. And there's many of them, and perhaps they mostly arose from non-genic sequences, de novo, creating entire molecular innovations. The human genome has a number of de novo genes that are arisen since the split between chimpanzee and human. How many are there? Don't ask me. I don't know, because there's been many reports that all have a different answer. All of them say there are some. The numbers range between 18 to over 600, and more recent papers say even more. The answer really depends on what you define as a gene, and like empirically, how do you say a sequence is a gene? And empirically, how do you say it arose de novo? So this is a young field without a consensus, except that they exist. And most of them are newly discovered and we don't know what they do, but some of them increasingly have been associated with disease. Because the question is at the heart of how we find de novo genes and how we study de novo gene emergence, I want to spend a second discussing, okay, what is a gene? Those of you who work in molecular biology may be familiar with this question. It does not have an answer. It is a hugely debated historical question because we learn what is a gene as we learn how genes work, and it changes. But for the purpose of my presentation today, we're going to have this very restrictive definition. A gene is a discrete DNA locus that codes for a functional protein. So with this definition, genes correspond to discrete loci scattered along a chromosome, and they have a clear beginning and end, each of them. And in contrast, every genomic sequence in between, we call it a non-genic sequence. That can include regulatory regions, that can include non-coding RNAs, that can include repeats, just not a gene. It's quite straightforward to describe a gene. It's got basically a coding sequence. So a sequence made up of triplets of nucleotides, codons, between a start and a stop, and that's called an open reading frame. And some open reading frames code for genes. And then I drew here some, meaningless ATG. You can have other open reading frames in the genome, but we don't think they code for genes. What differentiates the open reading frames that code for genes from the other ones, in theory, conventionally, is that their sequence is information that is read by cells through the processes of transcription and translation to generate proteins. The gene sequences are read into RNAs that exit the nucleus and are then translated by the ribosome, the cellular machinery, through the process that understands the gene sequence with the transcript sequence through the genetic code, which associates triplets of nucleotides to specific amino acid. This generates the primary amino acid sequence of a protein, which then can fold into all sorts of ways that give a protein its function, its structure, what it can interact with, say, with the membrane for a channel, or with metabolites, with DNA, and then it can do something. So that was a recap and really represents conventional what we think. And because of this well-established principle of molecular biology, since the early days of trying to find genes in the genomes, we use this principle and what we learn about the sequence patterns of open reading frames that encode proteins to kind of guess where the genes are in the genome. This has been done for 20 years, still being done today, but there is now a new way. In the omics era, we can interrogate experimentally, directly, what in the genome is translated. And that is thanks to a technology called ribosome profiling, or RiboSeq, which consists in basically deep sequencing the cDNA that corresponds to the RNA that was protected by the ribosome from nuclease digestion. In brief, finding which pieces of transcripts are in the ribosome and therefore being translated with bioinformatics treatment. This technology was developed in yeast in 2009, and very rapidly after its initial development, it became clear that there was pervasive translation of many open reading frames that we thought were in non-genic regions. They have been initially found in yeast and then found in many other species. And we call them, you may have heard of them, we call them non-canonical ORFs. And non-canonical means we didn't know that they existed. We didn't know that they were translated. That's what it means, really. So in yeast originally, now throughout humans, plants, mice, flies, fish, everywhere you look, you find these non-canonical translation events. But the physiological relevance of this non-canonical translation is unclear. One reason for this is that it's not very reproducible. This is a bit of a complicated figure, but it really summarizes an effort to just consolidate what has been reported to be translated outside of known canonical genes in the human genome, leading to a consensus set of 7,000. In this paper, we call them RiboSeq ORFs, but they're non-canonical ORFs. They're just new translated elements, newly discovered. And what you can see is that out of the 7,000 or so, 4,000 or so were detected in only a single study. So what is this? Is it relevant? A super recent paper that came out on Sunday showed that these newly discovered translated elements have biophysical capacities for interacting with canonical human proteins. This is again a complicated figure, but the idea is that they started from these 7,000 ORFs or so and used a membrane-based in vitro co-immunoprecipitation assay to interrogate what fragments of these small ORFs might bind to from a cell lysate. This figure shows like, okay, for each of the proteins that they assessed, what kind of evidence did they have that these proteins are real in vivo? And you see it varies a lot, but nevertheless, they find a lot of physical interactions and further study them. It seems that perhaps there's binding to domains that are shared across multiple proteins. So there is this translation, and it gives these kind of basic biophysical properties of small, tiny proteins that binds to things. What is the physiological relevance? My hypothesis is that these elements are what I call protogenes involved in the process of de novo gene birth. And this is kind of represented in this schema, where what I propose is that the translation of non-genic sequences gives sequences that are no longer non-genic because they're translated, but they're not quite genes because they're just random peptides. And often they bind to things, I don't know, but they're short-lived and they disappear. And occasionally they are retained by selection and can become genes, which then can evolve through the processes of descent with modification that I described. And this model is really a copy, a reverse copy of something that has been well known for a long time, which is the process of the death of genes, where a gene that existed can accumulate mutations that make it what we call a pseudo gene. So it's no longer working as a protein, but it can come back and resurrect if there's new mutations, or it can just disappear into the non-genic space. So this is what I propose for gene birth. And this is what we the field has established for gene death, very similar. For gene birth, a bit more specifically, this is more the mechanism. Like the idea is that in non-genic sequences, there is a lot of hidden genetic variation. Because non-genic sequences don't code for functional proteins by definition, they are not under the type of selection that prevents mutation from establishing themselves. So there's really just a lot of mutations that happen. And when some of these mutations are exposed by translation, it just creates a very large reservoir of protogenes, which most of it we expect be evolutionarily transient because the proteins encoded by the protogenes are not particularly useful. Perhaps they're even toxic. But occasionally, selection can take hold, perhaps when there's a change in the environment, that there's molecular novelty. And when that happens, we think the underlying locus can become under a different kind of selection and then progressively evolve the properties of genes, in particular, increasing its expression level, for example. And so the idea is that evolution would exploit dispensable sequences to generate adaptive innovation. So my lab, we study this idea using the budding yeast Saccharomyces cerevisiae as a model organism. Far from health and disease, but the best for understanding mechanism. The yeast genome was sequenced in 1996. It was the first eukaryotic genome to ever be sequenced. And by analyzing the sequences at the time, it was found that the yeast genome probably encodes 6,000 genes. And using these more omics ribosome profiling analysis that we've developed some computational methods that I will not tell you the details about, we find that in addition to these 6,000 genes, the yeast genome translates approximately 18,000 protogenes. This is, a huge change in what we think about how the cell works. Because there's so many new translated elements that are not only new because we just discovered them, but they're new because their sequences are unique. This is an example of a protogene. So you can see how we find them and how we find these 18,000. This is an open reading frame. It's unannotated, it's in the non-genic regions. It has a start codon, you can see here. This is its sequence in the budding yeast. And at the end, it's somewhere, it has a stop codon. On the y-axis, we show the ribosome profiling reads mapped at a single nucleotide resolution to this sequence. And we color the bars corresponding to the number of reads per nucleotide according to what position in the codon they are. So black for in frame and green or yellow for out of frame. And what you can see, I think, pretty visually, and we have, of course, statistical ways to assess that, is that there's a lot more black than the other colors, meaning the ribosome is progressing through this sequence in a triplet by triplet manner, which is the characteristic of translation. We don't know any process that would lead to this periodicity of read mapping except translation. So this is enough. It is translated, but it also newly emerged de novo from a previously non-genic sequence. Then you can see this based on the absence of ORFs in the orthologous DNA sequences aligned here. These are closely related yeast species that we can align by synteny very well. You see that the alignments are good. This is clearly the DNA has evolved by descent with modification, but the coding potential emerged somewhere in there. It wasn't there at the root, the common ancestor of this species. And the protein sequence coded by this open reading frame is absolutely unique to the species Cerevisiae, which is most evident if you look here. There is an out of frame shift, you see? And so that means that when you read 3 by 3, the sequence, like the ribosome would do with the genetic code, you read an entirely new sequence in Cerevisiae compared to the other possible species. So this is a protogene. And there's so many thousands of them. They exist in the cell. Might a protogene like this encode a functional protein? In other words, is it a gene? How can we tell the difference if we translate it? Well, it's based on these evolutionary analyses that I just showed you. So the principle is that DNA sequence codes for protein sequence, which itself encodes protein function that we just reviewed. Mutation that cause frame shift change the protein sequence drastically. And if that changes protein function in a way that has any importance, a mutation like this, a frame shift, should be selected against. This is because, again, of the genetic code. So we say that offs that tend to be purged of frame shifts and retain their frame, their integrity over evolutionary time are under purifying selection, which is, you know, established for 100 years of evolutionary theory as a clear evidence of function. And we don't see that in the protogene I showed you. And we don't see that in pretty much any of the 18,000 protogenes. They're all riddled with frame shifts. And yet, and this is where I start needing your help thinking about this, we are beginning to establish experimental evidence of protogene function in vivo. This little drawing here represents a yeast, for those of you who are not working with yeast. This is what it looks like because it buds. We compare how the yeast grows when this protogen that I showed you is here with its normal start codon, and when we make a single nucleotide change to the entire genome, and that nucleotide is transforming the start codon ATG to AAG. And we then also have another yeast where, in addition to making that change, we just express the protogene protein heterologously from a plasmid. These three curves show you the result of this experiment, each in three different just experimental conditions. What do we put in the media where the yeast grows? The black line here is how the normal yeast grows. The purple line is what happens when you make this one nucleotide change. The only difference between the black line and the purple line is a one nucleotide change that disables translation of that protogene. And what you see in the three different conditions is a drastic reduction of the growth rate of the organism. The dotted purple line that you barely can see because it pretty much overlaps the black line is what happens when you take these sick cells and you add back the protogene heterologously. This is it. And so what you can see is that blocking translation of this protogene is very deleterious to the organism, and that effect is fully rescued by adding the protogene back. Some of you are probably very familiar. There is nothing except in the technique of mutagenesis that I don't go into. There's really nothing crazy about this experiment. It's very, very classic loss of function and complementation experiment, which is the gold standard for establishing the function of a gene. So here we have the gold standard function in vivo and not in evolution. This is a problem. Like, let me explain why this is a problem again. Like, the concept, again, ingrained since, I don't know, this is like all of my undergrad was about this. From DNA sequence to protein function. Experimentally, in vivo, we can see it. But evolutionary theory over 100 years tells us that frame shifts should lead to purifying selection in a functional proteins. And we do not see this. This means to me, okay, what is the meaning of function? And for those of you who are interested in these questions, which I am interested, I collaborated with a philosopher and a rhetoric scholar to think about what function means in the case of de novo gene emergence. When there is nothing and then there is something, what is function? How do we think about that? And long story short, thinking about this problem leads me not to an answer, but at least to a framework of how to think, to work on it for now. And our approach going forward is to really study independently all of the different components that make a gene or not for these novel protogene elements. Like separately, like how are they expressed? What do they have a structure? What do they look like? Do they localize? What do they do? How do they affect phenotype? How are these phenotypes affecting fitness, etc.? The idea behind this being that we may see things that are expressed, but that's it. Maybe they're degraded immediately. We may see things that have signals and structures that lead them to somehow be in the cell and bind to things, but with no impact on phenotype at all. Who knows? We don't know the answers. We're really just trying now to observe. And I just want to show you like our poster child that we're working on. It's another yeast sequence that basically we entirely reconstructed its evolution. We performed ancestral sequence reconstruction for the first time in a de novo emerging locus. And we found, that it came from a non-genic sequence that didn't have any coding potential. We pinpointed exactly when the coding potential emerged and followed it through every branch of this small tree of yeast-related species. And the protein encoded by this protogene is predicted to have a transmembrane domain, and we verified this experimentally with carbonate extraction, etc. And in fact, they are in pink, and what you see is that its homologs, its ancestors also are predicted to have a transmembrane domain. And then even the non-genic sequence it came from and the places where it stays non-genic, what we did is we fake translate in the computer these sequences that are not coding, but we pretend they are. And if you do that, you'd see that the code hidden in there would encode for a transmembrane domain. And in fact, we figured out why. Let me tell you. It turns out that any random sequence that is rich in the nucleotide thymine has a huge propensity to make a transmembrane domain because of biases in the genetic code. What we see, looking more in details at the transmembrane protein, is that at the time of emergence, it's predicted to have a transmembrane domain, but looking more in detail, there are helix breaking residues that make it basically an unstable, like between a helix and beta sheet. We cloned this reconstructed element. So we just made a sequence that what this looks like, and we put it into cells, and mostly it just like aggregates places. But in some cells, you can see this tiny, tiny little ring here. And that's the membrane of the endoplasmic reticulum. Sometimes it integrates there. When we clone the next ancestor, it integrates there just fine. And this propensity to integrate the membrane of the endoplasmic reticulum is retained in these two descendants of this ancestor, in Cerevisiae and in Paradoxus, shown here, but not in this lineage where it has been lost. And evidence suggests that there's positive selection happening there. And we are actually currently working very hard to understand the mechanism of what this new protein does in the endoplasmic reticulum. We can see phenotypes, we can see interactions, and we're going to figure this out. But for now, I just wanted to show you how we are trying to think about this, which is basically just separating very conventional questions instead of merging them together. And that really brings me to discuss with you, like, what are the implications of these discoveries? So, you know, what is a gene? Like, yes, in theory, it's a discrete DNA locus that codes for a functional protein. And in theory, there are genes and there are non-genes, random sequences. In practice, it's never been easy to tell the difference. In the 25 years since the yeast genome was sequenced, some sequences that we thought were genes, in fact, we found out they looked like genes, but in fact, they were not. Some actually, they didn't look like genes, but in fact, they were. So this is why every year genome annotations change and change and change, because it's hard to tell the difference. And really, the idea emerging from our research is that there's more of a continuum between non-genes and genes, with protogenes in the middle that have some but not all of the characteristics of the protein coding genes we know and love and have studied for decades. They are transient entities, and they really seem to be evolving under different forces. Again, so my research in yeast is very advanced mechanistic. So it's not very advanced, you saw the bulk of it, but it's more advanced mechanistically than the research that is ongoing in parallel in humans, of course. But the existence of protogenes, I showed you, the fact that they can, you know, be in the cell, potentially do things, that is clear. That the protogenes themselves have novel sequences, but their existence appears to be ancestral. So in other words, the conventional model is that by descent with modification, evolution happens when there are changes in the sequence and copy number and regulation of genes. But what could be suggested, but what we as a field are seeing, is that also today there's all these other extant transient proteins, the protogenes, and that they are entirely completely different in their identity, in their sequence, from some that we do not have access to, but that likely existed in the ancestors. In other words, because the way proteins influence phenotype is really by interacting with each other and with the environment in the cell and external as well, it's like the ancestors' molecular network had the ancestors' genes, and they participated in phenotypes, but so did this entire world of small proteins we may never have access to, some of which perhaps are still here in a de novo gene form, but we also have a ton of other things. And so that we must account for to understand how the cell works and how health and disease work and differ between individuals and populations and species. So these are my food for thoughts for you. Thank you for inviting me again. Thank you to the lab members, collaborators, funding. I will highlight that a lot of these work and a lot of these thoughts are from Nelson Castillo in my lab and Aaron Weisholder in my lab. And so thank you to them and everyone else. And I will now be very happy to start the discussion, which I've been looking forward to.
Bravo.
Thank you.
So you have a couple of really good thoughts in the chat to start, and I think I'll call there first. So Neil Greenspan, would you kindly bring up your point first?
So in immunology, it's been known for several decades that there are pseudogenes in the immunoglobulin gene loci that are not directly transcribed and translated, but they are donors of sequences for the genes that are transcribed and translated. And so portions of those sequences ultimately can end up in functional antibodies,
Yes.
even though the actual segments from which they are derived are not normally thought to be directly transcribed or translated. And a very interesting, although perhaps one-off phenomenon that was described some years ago, not that many years ago, by Antonio Lanzavecchia and his group. They were looking at malaria antibodies, and they found an antibody that bound a particular family of parasite-encoded proteins. And when they sequenced it, they discovered, and this was entirely novel as far as I understand it, a sequence in the variable domain that had been donated by a non-immunoglobulin gene. And to my knowledge, that is the first time anyone has ever seen a gene conversion event between a non-immunoglobulin, I think it was even on a different chromosome, into an immunoglobulin locus, which is pretty bizarre. And it conferred excellent binding. Another point I would make just very quickly is that from an evolutionary perspective, a gene could be defined as or characterized as any sequence that when mutated changes phenotype. And I mean, even before that, we commonly talk about rRNA genes, and they don't encode a protein, they encode an RNA to make proteins.
Yeah.
So I think essentially where that is all going is to the notion that gene function is a polythetic concept. There is no one single definition, and you don't have to have all of a set of attributes in order to qualify.
Yeah, thank you very much for these comments.
And there's a number here in the chat. So David Rasmussen, are you on? Did you?
Yeah.
Oh, great, perfect.
I was just wondering, Anne, do we know anything about the relative risk of these translated proteins forming misfolded protein structures in cells that could cause disease? Like I was thinking of like amyloid plaques, but I guess there's a number of ways in which misfolded proteins could cause disease in cells.
Yeah.
And also, now that I'm thinking about it, would that generate sort of a countervailing selection pressure against these misfolded proteins that might sort of be a reason why they get quickly degraded if expressed?
Yeah, excellent question. So do we know? Very little. Something that I didn't mention, but I find very interesting is that studies of human de novo protogenes and yeast de novo protogenes report extremely different biophysical capacities. And a couple studies have tried to figure this out. And what might be is that the nucleotide composition of the non-genic sequences of human, which evolve completely differently than yeast, which has a very compact genome and a different GC content, they have a tendency to make more disordered domains, disordered protein domains, versus in yeast and possibly in other microbes, we have more hydrophobic, potentially globular and transmembrane domain. So the first thing is that we don't have an answer to your question, but it could be a lineage-specific answer. We don't know. However, I will say something important is that when translation happens, it makes a polypeptide. This is the only way translation works. But what happens to it is entirely unclear. So there are examples, we see the protein is expressed. But for the vast majority of these translated elements, we don't see the protein, at least not with conventional shotgun mass spectrometry, in part clearly, in fact, we don't have the power to see them because they're too low expressed, tissue specific, etc. But also perhaps they're just not there. Perhaps, as you suggest, they're made and degraded immediately. They're proteasome food because they would be toxic, etc. That's possible. Now, something that's extremely relevant to your question is that this process of just call it non-canonical translation. I talk about it in evolution over deep time. In fact, it also happens and changes somatically. So there are somatic mutations or events that yield, we call it aberrant translation events. So like too much elongation or a link RNA is now translated. And this has been in very few cases associated with disease phenotypes in models, in organism models. And a lot of the products of these, both of those, the somatic and the germline, I guess, translated elements, have been found in the MHC. So that's different from aggregating, and maybe they do aggregate, but we don't really know. But there is evidence that they are involved in health and disease somehow.
Thank you.
Cool. So there's one quick one from Lynn Fellman. Are you using long read technologies to sequence any of these regions? Yeah.
Yes. And I cannot talk to you too much in detail because it's a mess. It's a mess. It's crazy. If you look at one region, it can be in, I don't know how many different transcripts. And a transcript can map to, I don't know how many offs. So we are using the long reads. It's fun. We're concluding that we cannot actually conclude anything from it because what we need to know is what transcripts are translated. So we want to, but we haven't yet, do long read coupled with ribosome pull down to actually figure this out.
Super. So Dan Schreider, do you wanna pipe in?
I was mostly just responding to David's earlier question about how it'd be tricky to find evidence of deleterious ones. Looking at somatic protogenes is a great way to sort of get around that. But in terms of evolutionary timescales, they would disappear very quickly, right?
That's the expectation.
Yeah.
Yeah.
So Jonathan Parr, are you on? Your comment, your question's a broad one. Great.
Sure. Yeah, and I think you've given a couple answers have come up in the discussion, but my interest was piqued when you said you collaborated with a philosopher on the question of function. And I guess a follow-up question for me was just if you could give some more examples of what different forces may be driving the evolution of protogenes. And I was kind of asking at a higher level, like to benefit the protogene itself, to benefit the cell, to benefit the organism.
Yeah. No, totally. So I love it. So your guess is as good as mine, right? Like we are literally at not an impasse, but a place of all the hypotheses need to be asked and tested. The ones you suggested are very fun. Why not? I'm thinking of other ones. So for example, because we saw that it's very easy to make transmembrane domains by chance, what if we had genetic redundancy? So what if there is always a pool of transmembrane domains and it doesn't really matter exactly where they're expressed from? If you lose one, there's another one. And so maybe redundancy could be driving some of this rapid evolution. So maybe also you can have something that appears and binds and it's a bit deleterious. And so there is a compensatory mutation in a gene somewhere. So now everything is good. So you know what I mean? That's a possibility. An exciting idea that I have, and I have zero evidence for, is what if these are responding to rapidly changing selective pressures? What if the timescales that we think of are just not relevant to these elements, and they just evolve in response to selective pressures that are going at the scale of actual individuals and populations? So maybe in the summer and in the winter, you have different elements translated. Who knows? Yeah, I mean, I have many other complete theoretical ideas, none of them tested. And so I like the one that you said, like, is there selfishness in the elements themselves? Could there be regulatory genetic conflict?
So I just want to, I think on the same topic, I think Cynthia Beale's question is a nice tie into this. She asked, how long did the transcripts last?
The transcripts?
Yeah.
Oh, I'm gonna need to ask for clarification. Are we saying like what's the half-life of the transcripts inside the yeast cell, like from transcription, how fast are they degraded inside the yeast cells? Or are we asking how long in evolution is a region transcribed?
Within the cell.
Okay. I don't know.
Yeah.
Sorry. I wish I did.
Okay. Edmund Legrand has had his hand up for a while.
Hi, yeah, this is, let's see, get the video on, wait a minute. Okay, this is sort of mind-blowing, and if I've got it right, with the yeast, and I want to go also to an animal, for instance, but in a yeast, am I understanding it right? You've got your normal 6,000 genes that are doing what they're supposed to do in a well-functioning cell, and now you've got hundreds or thousands of other little proteins running around, sticking everywhere, and
tens of thousands. We have 18,000, which is not hundreds of thousands, but it is a lot more than 6,000. Yes, you're correct.
it's like somebody running around your house, putting nails all over the walls, and most of them are not any good. Most don't do anything, and once in a while, one is really good. So
We don't know.
it makes sense that in that yeast cell, if that good one happened, it would, it would replicate faster and be preserved. But in a, say in an animal, when you've got one germ cell, I wouldn't have wanted to take a chance with my, with my zygote for that type of risk.
Yeah.
So I should have less of that uncertainty because I've only got one shot at life, whereas a yeast population.
So that's a very interesting thought. I want to comment because there's been some excellent work done in Drosophila models looking at basically in the reproductive cell lineage. So apparently in the sperm development, there is a big relaxation of the chromatin state, and there is just actually a ton of pervasive expression. So there's actually more of those possibly in the sperm. And in a paper last year, Li Zhao from Rockefeller showed that one of these kind of pervasive, potentially random element actually provided an advantage. I don't remember the details, but it was either a sperm competition advantage or it was something like very, very close to the reproduction event. And so she proposed, based on little but exciting data, that this is actually the best spot. Where there is sexual reproduction, this may be the best spot for innovation because there is the strongest impact.
And if this showed up in, say, one of my hepatocytes, if it made it worse, it's no big deal. It could be replaced. If it made it better, it doesn't make much difference because it's just one cell and it can't be transmitted anyway. So there's not much advantage to having this kind of, I'll call it unregulated peptide synthesis in a soma.
Yeah,
That makes sense?
I agree. And so there's actually this raises two excellent points that are thought. So one is that I thought it was unregulated, but I don't think it is anymore. In fact, we see clear evidence of regulation. It's actually a regulated process. I don't know what regulates it and why, but it's very regulated, it seems, in fact. And so in the few examples that have been linked to disease can be thought of as a dysregulation problem.
But even in the yeast, there might be certain, in an individual yeast cell, of these many thousands of extra ones, there may be some of those peptides that are made in large amounts and
Yes.
others in small amounts.
Yes.
And if it's a good benefit in the large amounts, then that would tip it over the threshold and affect survival. Whereas if it was a good one, but only one peptide ever made, it might not be able to make a difference.
Yes, yes. Our data is consistent with this idea, but we don't have the sample size to be certain. But it seems that this might be the case. And this would also tie in with the idea that increase in expression level are part of the further evolution of those that are quote unquote good.
So I want to give Bernie Crespi, who's also been patiently waiting, a chance to ask the last question. I want to apologize to everybody else. There's a bunch here and you did a great job of stimulating a lot of questions.
Thanks for that. I just had a question about possible similarities between the processes that you've described and what presumably went on during the early evolution of protein-based life forms when there would have been much in the way of quote unquote experimentation and the evolution presumably of many de novo genes. And then if there were similarities with the origin of protein-based life forms, could there be any vestiges of those left in primitive primitive organisms that one could work on?
I love this question. I mean, as you can guess, I do not have an answer. I have no idea. But what I will say is that we did, like in our experiments in yeast, the frequency at which we observe beneficial effects, either through loss of function or gain of function, which I didn't describe, it's much higher than what people see in random library screens. So that means what the cell is trying out is not purely random. The genome is not purely random. And so I don't know how it was at the origin of life with what amino acids were available in the environment, but there is something not that trivial in this reservoir and turnover idea. And
Thank you.
of course, no, thank you for your question. I wish I had something to say about the origin of life, but I really don't.
Well, I suspect it might be worth a look. In this regard, it might be worth looking for parallels potentially.
I agree. I completely agree. And because Vaughn said yours was the last question, I just want to tell everyone else to please email me. I want all your questions and ideas.
Okay, fantastic. Thank you, Anne. This was a fantastic Club EvMed. There's so many great directions to go with this work, and I'm really glad to have an introduction to it and to hear about all these different dimensions of this work. Thanks so much for participating in Club EvMed. Vaughn, thank you for hosting. Becca, thank you for all your help setting things up, and thanks to everybody for tuning in.